Now I'm going to show you how to count human neural precursor cells, and you would count your cells when you want to play them for immunocytochemistry or when you want to do a transfection with nuclear factor machine. I use a phase contrast hemacytometer that has an edged grid, and also we use a dye called trypan blue, and the dye is useful in that it can differentiate between live and dead cells. Dead or dying cells take up this dye and they appear blue when you look at the cells in the hemacytometer, and live cells are able to exclude this dye and they don't appear blue. So that way you can determine the viability of your cells. To prepare a solution of trypan blue and media, I'm going to add 20 microliters of trypan blue solution into this 0.5 ml